right, ladies and gentlemen, third time is hopefully the charm, as this is the third time I'm trying to record this video. We're going to learn some details about what a chi-square analysis means, what we use it for, as well as go through some practice examples, and as this meme shows, how to use it as a pickup line. Warning, that will probably only work on nerds. All right, here we go. So what is a chi-square analysis? Basically, you are making predictions. That's what a hypothesis is after all. And you're trying to figure out, are your predictions statistically different from your observed results? When we were talking about probability in class, we talked about the fact that probabilities aren't certainties. Just because I flip a coin, even if the coin is perfectly fair, there's no guarantee that exactly 50% of the coin flips will be heads and exactly 50% will be tails. It's just there's a 50% chance or an equal chance that a coin flip would result in heads versus tails. So if we say flipped 100 coins, and we got 52 heads and we got 48 tails, we'd be like, oh, okay, that probably works out to, pretty close to what probabilities would predict. On the other hand, if we flipped a coin and we got 10 heads and we got 90 tails, we might start to think, there's probably something else going on here besides just the random chance. But where do you draw the line? For example, what about a 60-40 split? What about a 70-30 split? How far is too far from your expected results to be trustworthy? And that's what a chi-square analysis is for. So there's several topics on the AP test that can use chi-squared analysis. One is genetics problems. That's what a lot of your practice problems are, all the ones you have right now. When we get into Unit 7 with evolution, we'll talk about Hardy-Weinberg equilibrium, and we'll do chi-squared with that. There are several behavior labs that you can do, and we'll definitely do at least one free response question with that. And then you can also do the same thing with mitosis labs if you're trying to predict, for example, what percentage of cells that you observe in a microscope would be in a particular phase of mitosis or be in mitosis versus interphase. So in order to perform a chi-square analysis, the first thing you need to do is you need to create a null hypothesis. So a null hypothesis is basically your statement that the observed values are not different from the expected values, that your observed values match what you expect. What this is showing is that there's no outside force acting on this. If you're doing like a kind of classical scientific experiment, it would be, okay, there's no relationship between the two variables that I'm testing. If you're doing an analysis of whether or not a die that you've been rolling, a pair of dice, are fair, then that would be your null hypothesis would be A. The results we're getting, our observed values, work with the probabilities. Any difference from the expected values are due to chance. What we tend to most often think of as a hypothesis that we use for an experiment is generally called the alternative hypothesis. This is usually going to be stating what the researcher thinks is causing this phenomenon. And if you're confused about the difference between these two, we're going to look at some examples on the next slide. So if a null hypothesis has been rejected by a data, or it's not supported by a set of data, then there's going to be some sort of statistically significant relationship between those variables. So let's say that we're running an experiment. We want to see if club soda affects plant growth as compared to normal water. So where you basically take two groups of plants. One group is going to get watered with club soda. The other group is going to get watered with plain water. And you measure the growth of the two plants and, see if there's a difference. Your hypothesis might be, you think club soda would probably improve the growth of plants. So your alternative hypothesis is there's a relationship between these two variables, what I'm giving the plants and how they're growing. So that's if one plant is fed club soda for 1 month and another plant is fed plain water, the plant that is fed club soda will grow better than the plant that is fed plain water. However, a null hypothesis would be stating that there is no relationship between these two variables. Basically, it doesn't matter if you give club soda or if you give water to a plant. It's not going to affect the growth. So a way to state that would be if one plant is fed club soda for 1 month and another plant is fed plain water, there will be no difference in growth between the two plants. So your null hypothesis is there's no relationship, everything's due to chance. Unfortunately, for those of you who are not friends of math, there is an equation that goes with the chi-square analysis, and it looks a lot more complicated than it actually is. So, chi-squared is its own, separate variable. By the way, it looks like an X because that symbol is actually the Greek letter for chi, which was the X in their alphabet. Chi-squared is its own value that you calculate. It's not something you're going to take the square root of. And so this sigma here means sum. So we're going to be making this calculation multiple times, and we're going to add up all the results. What we're going to do is for each data set that we have, we're going to compare the observed data that we got and the expected data that we got. And so when we do this equation, we're going to get a value called chi-squared. If you look at your formula chart on the front of it, you're going to see a section called statistical analysis and probability. The only one of these equations that we haven't talked about so far is this chi-squared equation. Notice if you look on the right-hand side, the bottom 4 items all reference this chi-square equation, and those are all things that you need to be able to refer to if you forget what the chi-square equation has or what those variables mean. The third thing on here that you need is what's called chi-square table. We'll go over what degrees of freedom are later. We'll also go over what p-values are later. But just know that the line in this that we're really going to pay attention to has p equals 0.05. And I'll talk about why in a little bit. So let's talk degrees of freedom real quick. So degrees of freedom, first, before you can calculate that, you need to know how many possible outcomes can you have. In the case of a genetic cross, for example, between yellow peas, seen here, and green peas above, to the right of that, you have 2 possible outcomes. So your n is 2. Well, your degrees of freedom is going to be n minus 1. So in this case, it's going to be 2 minus 1. You have 1 degree of freedom. I will tell you that there is a method to the madness of this term. Degrees of freedom make sense once you kind of understand what it means. And I will tell you that the best explanation I've ever seen for this was on a blog that I found, and I'm going to put a link to it in the description of this video. So check it out if you want to have more than just, a memorized definition of what degrees of freedom are. So this chi-square table that you've got in your formula chart is important because it gives you what are called critical values. By the way, when I have them represented somewhere, I will refer to them as CV, but that's not like a generally accepted term. So if you're writing on an FRQ and you have to give an explanation involving that term, write out the words critical values. So we're going to compare the critical values in this table right here, and we're going to compare it to the chi-square value that we calculate. If chi-squared is greater than your critical value, you can say that you reject your null hypothesis, which probably means you're going to fail to reject your alternative hypothesis. If you want to say this in a way that would give a person who studies statistics hives, you can say, oh yeah, if chi-squared is greater than the critical value, then yeah, we go with our null hypothesis. No, we go with our alternate hypothesis. That's the one we think is really describing the situation here. If chi-squared is lower than the critical value, then it's our null hypothesis that's really accurate to our situation we're describing. But I will tell you this, on the AP test, in a statistics class, never say you accept the null hypothesis. It's technically impossible to completely prove a null hypothesis correct. So all you do is you say you failed to reject it. So on test questions, etc., when you're discussing things, I will count it wrong if you write except null hypothesis. So don't write that, but do write this. Okay? All right, so I've mentioned p-values. Now let's talk about what those are. A p-value is some number between 0 and 1, so it's going to most likely be a decimal, and it helps you determine the significance of your results. If you have a p of 0.05, that means there's a 95% chance that the difference between your expected and observed was due to something other than chance. And if you've ever calculated a p-value or are given a p-value and it's less than 0.05, that's strong evidence that you can reject your null hypothesis. If it's greater than 0.05, then that's usually weak evidence against your null hypothesis and you're going to fail to reject it. If looking at this and listening to this confused you, after I printed out your notes, I found this definition, which I thought was much better. A p-value is the probability that the observed results that you got, it's the probability of getting those results, assuming that the null hypothesis is correct. So if a p is 0.05, that means there's a 5% chance that you can get these results and still have your null hypothesis be correct, which means there's a 95% chance that your null hypothesis is incorrect. A smaller p-value means that there's more evidence in favor of the alternative hypothesis and less evidence in favor of your null hypothesis. If null hypotheses and alternative hypotheses are starting to give you a headache, don't worry. I had trouble with this very same subject when I was in AP Biology, only started to understand it better when I took statistics and college classes, and I still managed to pass my AP test. So let's do a practice problem. So we have homozygous green peas that were crossed with homozygous yellow peas. All of the offspring were phenotypically yellow. And this is going to be pretty standard of one of the genetics chi-squared problems that you're going to have. They're not going to tell you what trait is dominant, not necessarily going to tell you what trait is recessive, but hopefully they'll help you either give you the clues to figure that out or give you the clues to figure out the genotypes of the parents. So the parental generation is going to be, set is going to be homozygous dominant, the other set is going to be homozygous recessive. So we're crossing basically yellow with green. All right. All of the offspring were yellow. So the F1 generation, those were all yellow. No, they were all yellow, but they have to be heterozygous because one of these buddies has to be dominant, one of these has to be recessive, which means each of their offspring is going to get one dominant copy and one recessive copy. And I'm going to skip ahead, and we're using Y as the symbol for P color. So that means that one of these parents had to be homozygous dominant, and the second parent was homozygous recessive. What that means is that, apologies, the few times I will go back and erase, that the offspring had to be heterozygous. And if the heterozygotes are yellow, that means that the yellow trait is dominant because it prevents the recessive trait of green from showing up. So that means that this is the dominant phenotype, which is going to be important later on. Now, they say a test cross was performed with one of the offspring from the F1. That basically means, keep in mind these are plants, so it's not as creepy. They basically mated the plant with itself. They took the pollen from one of the anthers, and they used it to fertilize an egg cell from one of the ovules of the flowers. So ultimately, the parents and were the same individual, but you basically had a cross of two heterozygotes. And in A, they're saying, what are the genotypes of the parents used in this cross? Well, this is the cross they're talking about. That means that this is our answer. They're both heterozygous. So, if the F2 generation consisted of 20 offspring, predict how many will be green and how many will be yellow. So, remember our F1 was 2 heterozygotes. I'm not going to do Punnett squares because I want you guys to practice doing these genetics problems with probabilities. because once you get used to it, it can be quite a bit faster. So we have 3 different genotypes that you can produce in the F2 generation. So we can produce homozygous dominant, heterozygous, and homozygous recessive. If we are going to produce the homozygous dominant, there's only one way to do that. You got to get a dominant Y from one parent and a dominant Y from the other parent. What's the probability of getting a dominant Y from the first parent? Well, it's half. What's the probability of getting the dominant gene from the second parent? Also half. The probability of both of those events happening, getting the dominant allele from one parent and the dominant allele from the other parent, is found by multiplying the two. So that means that our probability of getting a homozygous dominant is 1/2 times 1/2, which is 1/4. We're going to do the other easy one next. So to get homozygous recessive, there's only one way you can do that. You got to get a recessive from one parent. You got to get a recessive from the second parent. You got a 1 in 2 chance of getting a recessive from parent 1, a 1 in 2 chance of getting a recessive from parent 2, and the probability of getting both of those is when you multiply them together. So that's another 1/4 probability. The heterozygote is a little more complicated. You got 2 ways that you can get that. You can either get the dominant Y, the dominant allele from the first parent, and the recessive allele from the second parent, or you can get the recessive allele from the first parent and the dominant allele from the second parent. Okay? Now, the probability of getting heterozygous, either way, is basically the sum of both of those. So 1/4 plus 1/4 is 2/4. And I'm not simplifying that to a half because it's going to make our math easier later. So let's think about phenotypes now. What color are these going to be? Well, we've established our dominant trait is yellow. So anything with the dominant allele is going to be yellow. That means these 2 genotypes right here. And our recessive trait is green. So this is going to be the only green possible combination to give us green. I would write in yellow and green, but I fear you would not be able to read it. So let's go back and think about this. Our total number of offspring is 20. Out of every 4 offspring that are produced, there is a 3 in 4 chance, because that's 1/4 plus 2/4, of having yellow offspring. There is a 1 in 4 chance of having green offspring. So what are we expecting? Well, we're expecting 3/4 of 20, or 15 offspring to have a yellow phenotype, and we're expecting 1/4 of 20, or 5 of the offspring to have the green phenotype. So this is giving us our expected values. And remember, in chi-square, it's all about looking at observed versus expected. Now, we have our expected values, so let's go ahead and craft a null hypothesis, because honestly, there's not room on the next slide for that. So among the offspring, the ratio of yellow to green peas will be 3:1. All right, we're going to keep that in mind when we're looking at the next step in this problem. So if the offspring consisted of 8, oops, too far. If the offspring consisted of 8 yellow peas and 12 green peas, is this statistically different from your prediction? So we have 2 different phenotypes. You can be yellow or you can be green. Our observed was we had 8 yellow peas and we had 12 green peas. What's the total number we had? Well, we had 20. That means our expected numbers should add up to 20 as well, and we already calculated those on the previous slide. That's 15 and 5. So then all we have to do is fill out this chart to do the math. On a lot of the AP tests that I've seen with FRQs, the ones that have chi-square problems, a lot of times they'll have some chart very similar to this. They may not break it down into as many steps, but they'll still have something similar so that it will help you do the calculations. But I'm going to go ahead and fill out the numbers. You feel free to pull out your calculator and do it as we do this, but 8 minus 15, because it's observed minus expected, is going to be negative 7. 12 minus 5 is going to be 7. My next column, I'm going to take the previous column's value and I'm going to square it. So negative 7 squared is 49. Positive 7 squared is also 49. Notice that I've blacked out the total column here, and I kind of already have something in this column. We're not calculating totals for either of those two columns. And then finally, what we're going to do in the last column is take that number 49, and we're going to divide it by the expected. So in the final column, it's going to be 49 divided by 15, which when you calculated it out is 3.27. Remember, I always round to 2 digits. Then 49 divided by our expected value of 5 is going to give us 9.8. Now this cell is open because I do want you to calculate a total here. You're going to add up those calculations, which in this case is going to give you 13.07, and that is our chi-squared value that we've calculated. Now we still have a little more work to do before we see if we reject or fail to reject our null hypothesis. So let's go and look at the next part of the table. So for our degrees of freedom, we need to know how many possible outcomes. Basically, how many phenotypes did we have? Well, we've got 2 different phenotypes. Our n is 2. Degrees of freedom is n minus 1, so we have 1 degree of freedom. If we look at our table for chi-square value critical values, we're going to find that for a p of 0.05, our critical value is 3.84. So our chi-squared value of 13.07 is greater than our critical value of 3.84. That means that we would reject our null hypothesis, because this inheritance pattern does not match our predicted phenotypic ratio. All right. D is asking you the same thing again, only now we have 10 times as many offspring. So we have the same things. We have 2 different phenotypes, yellow and green. And while we're looking at that, by the way, that was supposed to be degrees of freedom. This means our n is 2, so we have 1 degree of freedom. If we look at our critical values on our table, it's still going to be 3.84. All right, we have 200 total offspring. Okay, we have 80 with yellow peas, and we have 120 with green peas. Now we need to figure out our expected. If you remember from part B, we expect 3/4 to be yellow, and we expect 1/4 to be green. So for that, we would take our total number of offspring, and we're going to multiply it by 3/4. 200 times 3/4 is going to be 150. And, we could do the same process for the green. 1/4 of 200 is going to be 50. So then I'm going to go ahead and fill out the rest of the table. If you have math issues, you can always rewatch this, slow it down. Listen to me at super slow speed, although that might be too entertaining. And there's our chi-squared value, 130.7. How does that compare to our critical value? Well, our chi-squared value is super, super bigger than our critical value. That means our data really does not fit the expected phenotypic ratio, and we're going to reject the living daylights out of that null hypothesis. All right. I also have a bonus practice problem for you, just so that way on the chi-square practice problems, you do have some where you're looking at two traits at the same time. So hopefully this will make it a little easier for you to do those problems. So in certain strains of corn, the phenotype for color can be either yellow or purple, and the texture of the kernels can be plump or smooth or wrinkled. So those are our two traits, color and texture. A cross between a homozygous purple wrinkled and a homozygous yellow smooth was our parental generation. The offspring from this cross produced were all smooth and purple. So we had two double homozygotes that were mated with each other. I'm going to go ahead and just assign some letters. So for color, let's go ahead and say we're going to use the letter R. And for texture, I think that's texture. We're going to use H just because it makes it easier to figure out what's capital and what's lowercase. All right, so our homozygous individuals, which is what's produced in F1, are all smooth and purple. That means that both smooth and purple are the dominant traits for those the dominant alleles for those traits. And that's going to be important for when we're trying to figure out frequencies for our F2 generation. All right, we're going to do some more math now. Brace yourselves. All right, so our F1 generation, by the way, was our double heterozygotes. All right, so we're not doing the giant Punnett square. We're not going to do Punnett squares. We're going to do this with probabilities. So let's look at this cross. What is the probability of having purple offspring? Well, it's the dominant trait. If you've got two heterozygotes, they're always going to be 3/4 dominant, which is purple, and 1/4 recessive, which is yellow. Then we're going to go ahead and we're going to do this also for the second trait. And same thing, 3/4 of these are going to be smooth, because that's the dominant trait, and 1/4 of these are going to be wrinkled, because that's the recessive trait. And by the way, just to make my life easier, I'm going to write purple as P, smooth as S, wrinkled as W, and yellow as Y. Let's do this. We're going to write purple as PRP. Let's make that. Smooth is SMO, wrinkled is going to be WRI, and yellow is going to be YEL. Just because I've written this out a couple of times, and it's a lot of writing. So let's now look at our different possible combinations. We can be purple and smooth. We can be purple and wrinkled. We can be yellow and smooth. And we can be yellow and wrinkled. How are we going to figure out those probabilities? Well, it's fairly simple. If you are purple and smooth, you have to have this probability and this probability. What that means is there's a 3/4 chance of being purple, and there's a 3/4 chance of being smooth, and the chance of being both is the product of those two numbers, which is 9/16. If you're purple and wrinkled, that's this probability and this probability. So what that is you've got a 3/4 chance of being purple, and you've got a 1/4 chance of being wrinkled, which means your probability is 9 out of 16. To be yellow and smooth, that's going to be this probability with this one. So a 1/4 chance of being yellow and a 3/4 chance of being smooth, which is 3/16. To be yellow and wrinkled, that is a 1 in 4 chance of being yellow and a 1 in 4 chance of being wrinkled, which is going to altogether give you a 1 in 16 chance of being yellow and wrinkled. So these are our ratios. What this means is that these offspring should appear in a 9:3 to 3:1 ratio. And before we go and do anything else, let's go ahead and write out a null hypothesis. So our null hypothesis, let's say the ratio of, and I'm going to do my lazy writing again, purple smooth, purple wrinkled, yellow smooth, and yellow smooth, yellow wrinkled. That ratio will be 9:3 to 3:1. All right, so there's our null hypothesis. This is what we're expecting. Now, if you'll remember, we're going to have to calculate an expected value. So let's go ahead and do that calculation now while we've got some space and also while we've got some numbers worked out. So the first thing we need to do is we need to figure out the total. And let's write in black. So we need to add 375 plus 133 plus 118 plus 47. Our total number of offspring from this cross is 673. So to find out what 9/16 of that is, we need to multiply 9/16 by 673. If you do that, you get 379, and I'm choosing to round to the nearest whole number, but it shouldn't affect the outcome too much if you round to a decimal point. To find out what 3/16 of that is, of 673, you're going to multiply 673 by 3/16, and that's going to give you 126. Math is exactly the same for the next one, because it's going to be the same ratio. And then finally, to get 1/16 of 673, you're going to multiply those two together, and that's going to give you 42. And if you really want, you can do a final check and add them all back together, and you should get 673. If you had rounded and the numbers were a little different, you might have been off by 1 or 2, but like I said, shouldn't make the math too difficult. All right, so I have already gone and filled out a couple of things. Our phenotype. We have 4 different phenotypes, so that means, let's change the color back to the red. That means our n equals 4, which means our degrees of freedom is going to be 4 minus 1, or 3. If you want, you can go ahead and go to the chi-square table, and you can figure out what the critical value is at this point for degrees of freedom, for 3 degrees of freedom, basically. And it's around 7.815. I may have an extra decimal point on there. I didn't get this from the actual table on your formula chart, but it's still the same. So, our observed are the numbers that they gave us in the table. Our expected total is going to be 673, and our fractions are those numbers. So if we do the math, and I'm going to go ahead and just fill out this table. So 375 minus 379 is -4. 133 minus 126 is 7. 118 minus 126 is -8. 47 minus 42 is going to be 5. Square these numbers and I get 16, 49, 64, and 25. Notice though, when I get to the next step, the number I'm dividing by, my expected, is a really big number. What this means is that the larger the sample size you get, the easier it is for your chi-squared value to be a smaller number. So let's go ahead and do this. So we're going to do, for our first calculation, it's going to be 16 divided by our expected, which is 379, and that gives you 0.04. Next, it's going to be our number 49 divided by the expected, which is 126. When you do that, it gives you 0.39. 64 also is divided by 126. That gives you 0.51. And 25 is divided by 42, which is going to give you 0.60. Add all of those together and you get 1.54 as your chi-squared value. So your chi-squared value of 1.54 is less than your critical value of 7.815. This means you fail to reject your null hypothesis, which means your data does fit the predicted phenotypic ratio. So now you've seen one where you reject the null hypothesis, you've seen one where you failed to reject the null hypothesis, you've also seen 2 traits, 1 trait, which means you are now ready to go on to this. So this is the first question from your assignment. No, we're not going to do it together. I'm just pointing out how the different features we did fit into this. So you have a space right here for writing out your null hypothesis. I don't really give you space to do Punnett squares or probability calculations, but if you need it, you might have some extra space in here. You're going to write your expected phenotypic ratios. So an example, it could be like 3 to 1. It could be 9 to 3 to 3 to 1. It all depends on the information you're given up here, and it also depends on just the inheritance pattern. For each problem, you should have a data table with the appropriate number of rows. I'm afraid I have n written instead of degrees of freedom in that table. Go ahead and scratch it out and write degrees of freedom. You'll always be looking for the critical value on your chi-squared table. I don't have a space for you to talk about rejecting or failing to reject the null hypothesis, but when you reject it or fail to reject it, that's going to let you know if the data fit the predicted phenotypic ratio. And I do expect you to fill out this whole chart. Is it tedious? Maybe. But will it give you the practice in doing this and doing it correctly? Yes. And while you're only getting 8 practice problems, I don't expect chi-square to show up more than once on the test, hopefully, so I don't want us to spend too much time on this. All right, that is it. Next up will be non-Mendelian genetics, so be prepared.